Okay. With that, we would now like to welcome Dr. Paul Macklin for our second talk of the day. Dr. Macklin is an associate professor of intelligent systems engineering at Indiana University, where his lab develops open source computational tools to model multicellular biological systems. And his open source project PhysiCell enables large-scale agent-based simulations in three-dimensional tissues. His lab is especially focused on constructing models of cancer biology with the software they develop, but PhysiCell has also been widely applied to many other biological domains, including nanotherapy, microbiology, and immunology, among many others. In addition to this work, he and his lab have also been pioneers in developing data standards for multicellular biology, which will surely help encourage reproducibility in the domain. So welcome, Dr. Macklin. We're really glad to have you with us today and are excited for your talk.
Well, thank you, Veronica, and thank you, Herbert, for the invitation. It's always great to meet with you all, even if we only end up meeting virtually again. But thank you for inviting me to this wonderful workshop. I'm so glad that you're running it. So it's a great service to the community to do all this network modeling. And it's a great segue for us going into next week, because we're running our workshop on PhysiCell, and we will be directly integrating network modeling into our agents as part of our workshop, using both Boolean network models and SPML style models. So it's a tremendous synergy between our workshops. So we're really grateful for that. I will go ahead and share my screen here. Hopefully you all are seeing like basically too much shared, but I'm going to be switching between windows here. So I want to just share the desktop to make that easier. Can you all see the slide here?
Yep, we can see him.
Oh, fantastic. So part of the talk that we'll be doing today will actually use an online or a cloud-hosted resource showing kind of a cloud version of PhysiCell and a kind of, I suppose, a quasi-live demo. So we won't have time to kind of walk everyone through, but what I really encourage you to do when we're done is to download the slides and look at the video and kind of slow it down. You can actually repeat the modeling steps that I'll show you here today as part of the demonstration. And then next week, we'll be doing a whole lot more of this. And you can register for a free NanoHub account. If you go to nanohub.org/register, you can make that account. Just use your Google account like we do for everything, and it works out very nicely. So today I'm going to give you kind of a primer about using agent-based models for multicellular interactions, particularly using PhysiCell, which is our model system like Veronica introduced. We're really interested as biologists, as scientists, as mathematicians, in understanding how we can start with a palette of individual cell behaviors that they can divide, that they can grow, they can differentiate or change type, they can die, they consume things in the environment and sense things in the environment, they can secrete chemical factors. Of course, they can mutate and change type, and they can move around and crawl. And then you take those individual cells and they start to interact. They can stick to each other, they can push each other around, they can eat each other in phagocytosis. They can also transmit signals as they stick together through adhesion. And somehow you put this limited palette of single cell behaviors and cell-cell interactions together along with physical constraints in your environment, things like diffusion limits and mechanical barriers. And you put all that together and scale it up over thousands or millions of cells, and you get complex multicellular ecosystems. We see it in cancer, we see it in tissue biology, we certainly see it in immunology. And we want to be able to understand how these simple individual behaviors and interactions scale up to become the complex systems that we see in tissue ecosystems and other biological ecosystems. And that's what I would kind of phrase and term as multicellular systems biology. It's the study of these big multicellular networks of cells working in space and time. And of course, we don't want to just be scientists, but we want to eventually be engineers to take that newfound understanding of these systems and manipulate it to control those systems, to engineer a better tissue, to take a diseased tissue back to a healthy state, get rid of the cancer, get rid of the infection, get rid of the overactive immune response for autoimmunity. And so scientists use models to detangle complex systems. And us as computational scientists, we use computational or mathematical models. And so that's what we'll be building up as a computational model system today, using our agent-based framework and exploring a few problems. So what would be the key parts of a multicellular systems biology, virtual laboratory? Well, we know that chemical diffusing factors are really important drivers of cell biology. We know that there's exchange of growth factors and metabolites like oxygen, glucose, and growth factors, say glutamine and other things. We know that cells can secrete and sense chemical factors that diffuse through the environment. And then, of course, if we're trying to manipulate these systems, we also need to worry about therapeutic compounds that we put into the environment that diffuse and go wherever they're going to go. And then on top of that kind of like chemical environment, we have the cells working inside of it. They have environment-dependent behaviors, including molecular scale logic, like you're learning this week. They can, of course, interact mechanically. And the fascinating and exhilarating thing about biology is that there's tremendous heterogeneity, even within individual cell populations, that they can have individual states, individual parameter values, and individual biological rules or hypotheses. And so you want to be able to build up a system where you can model the chemical environment, model the individual cells with their individual rules as they interact with each other and their environment, and then to be able to run virtual experiments on that laboratory by running it in high throughput, and hopefully discover the rules that best match your observations. So you can really learn the cancer biology or the immunology and identify and exploit weaknesses that will help you to steer the system and control it as you want to. So kind of taking a step back, and you probably have already seen this, but an agent-based model is a discrete and stochastic model that models individual cells as software agents. They have their individual state variables, and they have individual rules. So it's like they have their attributes and their methods. And the rules can often be written as systems of ordinary differential equations, as Boolean network models, or other constitutive laws. But if you really take a step back, biological software agents in an agent-based modeling framework give us an opportunity to kind of directly encode our biological knowledge and hypotheses as rules for the agents. Then these agents will tend to live in a virtual tissue environment, which will often be modeled as diffusion problems with partial differential equations. And really, the environment is like the way of encoding the physical constraints on your cells. And then they interact with each other and this environment through the mechanics, contact, and other rules. And so you put those together, you get this hyper-discrete continuum framework where you have discrete cells that can have discrete events like division events, birth events, death events, movement or changes in state can sometimes be stochastic. And then you put it into that continuum model of the environment. So you get this discrete continuum representation. There are many approaches to building cell-based models. And we actually recently wrote a review, and JCO Clinical Cancer Informatics is open access, so you can access that for free, and we'd encourage you to look. And there are many ways to classify these kinds of systems. But one great way to look at it is to look at lattice versus off-lattice models. In lattice model, you divide space up into positions, and cells can occupy a discrete position. It can move left, right, up, or down by typically by fixed distances. And then you might say increase the resolution with the cellular pulse model by maybe say introducing more lattice per cell. But it fundamentally has these discrete jumps in cell position. Then you have off-lattice models where you say, let's not divide space up like that. We're just going to have, let's say every cell as an agent in space can have its own position. And then we'll talk to look more typically at balance of like exchange of forces to determine the motion of those cells. And so there are pros and cons for each of these approaches. And PhysiCell in this context is a center-based model where you have these off-lattice positions and the balance of forces. And so to kind of briefly introduce the parts of our virtual framework, the first part we model is the kind of the microenvironment using diffusion equations. So we built something called BioFEM, which is a finite volume method that has been designed specifically for these kinds of biology problems. So we vectorized as much as we could. We have a vector of reaction diffusion equations that we solve in parallel using OpenMP parallelism. And this scales up very nicely in the number of substrates and the size of the domain. And you can run this on a broad variety of platforms. It runs on Linux, runs on Mac OS, runs on Windows. You can develop it on your desktop, and then when you're ready to go, ship it off to a cluster and do investigations, all using standard C++. And so that's kind of the beginning part of our virtual framework of modeling that environment. And then when you go on, I guess one key part to introduce is that because we know this is biology, in these reaction diffusion equations, we treat the cells as point sources and sinks, so they can secrete things or consume things in the environment and affect the differential equations. And so that's kind of the environment. And then we built PhysiCell on top of that by saying, take those discrete point sources and sinks in the finite volume code, and now start adding in the biology of all the basic processes. So we added models of cell cycling and kind of that graph structure progressing through a cycle. We added death process models. We added random bias walks for motility. We added potential functions for adhesion and mechanics. And of course, an ability to modulate how much is secreting or consuming the environment. And we added a lot of now of the interactions, things like phagocytosis are all standardly implemented. And every cell agent in here then has its own copy of its own state, We have function pointers that will allow you to dynamically change the rules on a cell-by-cell basis if you want to. And then you can scale this up, and it scales order n in the cost scaling, so that works pretty nicely. And again, we worked really hard to make sure this is very self-contained and easy to install. And it works across platforms with no modification of your package. So you can have one team member who's developing on Windows, another member who's developing on Mac OS, another one on Linux. And when you're done with your code and you're ready, you ship it off to a cluster and run it unmodified. And so to show you a typical example, here is a cutaway view of a tumor. And what I've done here is to kind of show you this notion of heterogeneity. Every individual cell agent in this simulation is colored from blue to yellow. The blue cells are less aggressive. The yellow cells are more aggressive, so they have a higher proliferation rate. Then the other thing here is we have oxygen diffusion to the environment. So if you look kind of on the outside edges of this box, we apply a Dirichlet condition or fixed condition, and oxygen diffuses into the environment. Then the tumor cells in this particular example are consuming oxygen. So as you get deeper and deeper into the tumor, the oxygen level drops to develop an oxygen gradient. And if you get to the very center of the tumor, there will emerge a necrotic core, these brown cells where oxygen is dropped so low that there's basically energy collapse in the cells. So they start to shrink and stick and degrade over slow periods of time. And so watch, each one of these dots is a cell, and you can actually see the nuclei in this particular rendering. And you see how yellow cells give birth to yellow cells, blue cells give birth to blue cells. You kind of see the competition processes in this tumor as you go. Okay, here we have the necrotic core, so the oxygen is finally dropped low enough. And you see these patches of yellow clones of the most aggressive cells. Here's kind of an intermediate clone. And then the blue cells kind of being squished in between and pushed around. And so you can see that, very basic rules about oxygen-dependent birth and death, plus some basic mechanics, and the oxygen diffusion can lead to emergence of structures that replicate what we see in real life. So this is all published across platform. It's open source, and the entire subject of our workshop next week in teaching you how to use this platform. So what I'd like to do is to spend some time first with a couple example models to show you how basic interactions can be implemented in the cell agents and what kind of behaviors pop up. And so I'm going to kind of show a few characteristic and fun interaction examples. I would be happy to give more technical detail into the platform, but I figure that's kind of what we'll do next week. And I'd rather that we just have more time for fun, but I'm happy to answer at the end of the talk any questions about a little bit more of the technical underpinnings of the system and how it works. So for our first example, I want to just show kind of a classical example from synthetic biology, but now I'm making it a spatial temporal version of it. And so the repressilator is famous because it was like the first synthetic oscillator. And so you have like a network of 3 nodes, A, B, and C. And so protein A, when it comes up, inhibits protein B. Protein B inhibits protein C inhibits protein A. And so the way this works then, right, is if A goes up, that means B has to go down. But because B is down, it's no longer blocking C, so C can go up. But when C goes up, that knocks A down, which means A goes down and B can go up. And so you can kind of get this succession that, A, B, C, A, B, C, and you get this oscillator. It's like a natural clock. But what we're going to do now is we're going to make a multicellular version of this. We're going to have 3 cell types, A, B, and C. A's job is just to secrete A. But if A receives signal C, it will stop secreting. B will secrete signal B, as you might imagine, and it secretes it as long as it's not blocked by A. C secretes C unless it's blocked by B. So this is the multicellular version of this. And what I like about thinking about tissues and ecosystems is that you can take kind of a traditional circuit that you might use in multicellulars in synthetic biology or in other systems biology, and you can actually break that up over different cell types and have specialization, and you can turn that into a multicellular version of your circuit by having different cells play different roles. You can imagine, though, for the circuit to be complete is that they have to have A, B, and C cells close enough to read each other's signals. Otherwise, you might break the circuit where there's a big patch of A cells or a big patch of B cells. So anyway, let's see what this does. So I'm going to click on here. I'm going to color the A, B, and C cells based upon whether they're secreting. So if they're secreting, they're going to glow bright. And when they're not secreting, they're going to go down. It's like a fluorescent protein. And so you can kind of see as things settle out, we have these cells, and they're also proliferating, by the way, so you can see spatial effects, that we're getting this flashing between the different colors, just as we might expect, right? We have this repressilator that's supposed to be an oscillator. But as it gets bigger, the signals have to propagate by diffusion across the domain. which means that over time, as this thing grows, it can take longer and longer for the signals to propagate, and you have different parts of this colony in different parts of the circuit. And so you'll see now these waves of color changes sweeping across the tissue, and sometimes even getting the emergence of the classical spiral waves that you might expect in this kind of a circuit. So really simple rules on the individual cells, but you put it together and you get a multicellular circuit, which is like the simplest network you'll see this week. But you can put these into multicellular systems, and so that can be kind of fun to see these oscillations emerge. Another thing you can see in colonies of cells is a notion of specialization. And so let me give you an example. It's a little bit of a zanier example, but the idea here is that you have a whole bunch of cells that start off as individuals, and they're going to secrete a quorum factor to aggregate. So this is like bacteria, where the bacteria secrete a quorum factor, and then they use chemotaxis to move up the quorum factor to start aggregating. And so that's what the unattached cells are going to do. But now in the multicellular version of this, they're going to start secreting the quorum factor, and when they come into contact with each other, they're going to form spring-like adhesions and kind of try to form a long-term adhesion. And we're going to stipulate in this particular model that every cell has a maximum of 2 adhesion sites. So first part is just they're going to clump up, and they're trying to aggregate and form connections, and they can form up to 2 connections, which means they're going to be able to form chains of cells. Now, what we want to do is to be able to have some kind of specialization along that chain of the cell. In particular, I would like there to be a head end and a tail end. And so one way to accomplish that is to give every single individual agent has its own stochastically assigned rate of generating a signal, which we're just going to call the head signal. And if any 2 cells are linked, that head signal can flux between the 2 cell agents using a standard Fickian, Fick's law of diffusion. So what's interesting is you're going to have this chain of connected cells. Each one of them is going to secrete a little bit of that head signal at a stochastic rate. And so imagine you have the 2 ends now. Well, the probability of those 2 having exactly the same secretion rates is about 0, which means that one of them is going to have a higher value than the other. And so if you solve it, this is basically a linear diffusion equation, and one of the 2 sides is going to have a higher Dirichlet condition, going to have a higher value than the other. And so what the cells will do is they're going to compare their values to the neighbors and say, if I look, the direction where the signal is going up is towards the head, and I'm going to follow the directions of whoever's ahead of me. And that makes one cell the leader and as the head of this chain of cells, and one cell is the tail. And so with a very simple diffusion of a signal, like a juxtacrine signaling, basically across attached cells, you can get specialization. So let me show you what this looks like. So I'm going to first, at the very beginning, all the cells are gray because they're unattached, although I kind of made one here already. And they're wandering around by chemotaxis. Whenever they attach, basically they're going to start sorting out whether they think they're a head or a tail. So red will be a head tail, red will be a head cell, gold will be a tail. And in the middle, we're going to shade them from blue to white, where blue is going to be the lowest level of signal. So blue should be very close to the tail. All the way up to white is the highest value of the signal. So that should be close to the head. And watch as they kind of form up and form specialized roles, and then the head gets to form the leader, and that steers the chain of these connected cells, these little worms. So here we see that they formed up. The other one formed up. We have a clear head and a tail. Here we have a head and tail, and the head is kind of choosing direction and pulling the rest along for the ride. And so you can get these multicellular-like organisms, these worms, that emerge from very basic individual roles, and they don't have to have, omniscient knowledge to understand where they are in the structure, because you have this diffusing signal along the length of the connected cells. So just kind of an example of a toy model, but showing how simple individual cell behaviors with contact communication can allow for differentiation, specialization, and formation of coherent structures. Now I'd like to show you some examples a little bit more real, a little bit more research-driven. The first one's going to be a work about hypoxia or low oxygen breast cancer invasion. This is a work that's done jointly with Danielle Gilks's lab over at Johns Hopkins University and led by my postdoc Eva Rocha and her postdoc Anesh Godet. And this is recently published, I guess, last year in iScience. So you can access that as open access. So if you take, like, say, you plant a tumor in a mouse, and this is what Danielle does, and let them grow and kind of section them and stand them for different things, you see, just like the necrotic spheroid I showed you earlier, there's kind of an emergent structure, and you can stain for different things. So on the very outer edge of the tumor, these red cells have plenty of oxygen, they fluoresce red, and then you kind of get deeper and deeper into the tumor, and you get, let's say you go to the very center, Now, this region of dead cells, so this is that necrotic core that I showed you in my simulation. These happen in real life in vivo as well as in laboratory conditions. And so this is kind of the dead core. They've been stunned by tunnel assay. And then at the very edge, just before they die, we have cells that are responding to hypoxia by upregulating things like hypoxia-inducible factors. So these purple cells here are newly hypoxic. And then this particular system, if a cell stays hypoxic long enough, it switches to a green color. But the interesting thing here is that you have hypoxic cells here at the very back edge, but the green ones are a little bit forward. So they're doing something. They're migrating away to try to escape the bad conditions. And then you have a region of mixing between the cells with plenty of oxygen and the cells that have at some point been low oxygen. And so we know there's a phenotype change in the hypoxic cells. They change this more motile invasive behavior. And so the question that we had was, what about those green cells when they leave the hypoxic zones, when they flee the danger successfully? So now they have this newly hypoxic, newly motile phenotype, and they leave the signal that made them motile. Do they retain their old, their new motile phenotype and keep wandering forever? Do they instantly change back to their old normoxic phenotype and stop migrating? Or is there something in between, like a persistence time that say, I retain my old phenotype for a little bit longer? So we asked, so I went to a biology meeting about 10 years ago now, and asked that question basically to every biologist I could find in the room. I basically did an informal survey saying, if you take a hypoxic adapted cancer cell and you place it back into normoxic conditions, what will it do? Will it go back to its good old-fashioned normoxic behavior? Will it retain its old hypoxic behavior, or will it take some time to transition? And so I asked them, how long would it take to switch back? And the survey result is basically the entire right half of the real line. Some biologists say, no, they change instantaneously back to the old behavior. Other scientists said they will never change the old behavior. And I had every value in between 0 and infinity of how long it would take them to switch. So that was kind of fun. And it's clearly a right question. So Danielle developed this beautiful, beautiful system where the cells are engineered, the cancer cells are engineered to fluoresce red ordinarily. But if they've been exposed for enough time to hypoxia, they will switch up, upregulate HIF1 alpha, as you've probably seen in some of your talks. And that will basically cause them to snip out the red gene and permanently change color to green. So what this means is that you can track a cell that either is or has been hypoxic and see what it does. And allows us to answer these questions saying, when they leave a hypoxic zone, what are they doing? Well, we can tell if they've been hypoxic in the past. So this beautiful tracking system. And so we built a model to model not just the cancer, but also this tracking system. So we simulate oxygen diffusion and uptake. We made proliferation vary with oxygenation and with mechanical pressure. So if a cell is too squeezed, we'll downregulate pressure. Live cells in the normoxic zone are red, just like in the engineered system, or they are green if they've been hypoxic for long enough. And that's a permanent change in our system, along with basically we use an ODE system to model that change in expression. And then we said that green cell types have a migratory phenotype to move up oxygen gradients with a biased random walk. And then we added a phenotypic persistence variable saying that whenever they're out of a hypoxic zone, there's a probability of them switching back to their non-motile or less motile behavior. And so Aber did a great job of fitting this with experiments. He did a beautiful approximate Bayesian computing for parameter fitting to get the differences in the red and green cells. And then we applied them to model a spheroid. And so we explored this phenotypic persistence question, saying, first, let's do no phenotypic persistence. So that's the top row, saying that once a green cell escapes its bad conditions, it immediately switches back to its old non-motile behavior. And as you would probably expect, right, that you have no invasion of the system because the green cells will escape just long enough to reach bad conditions and then reach the edge of the bad conditions and stay put. And there will always be some red cells between them and the edge of the tumor that have never experienced the bad conditions. So you get this growing concentric ring structure of red normoxic cells on the outside with green migratory cells or barely migratory cells on the inside and the necrotic core in the center, and you never get invasion. But we know that's not true. Cancer does invade, unfortunately. So then we said, well, let's turn the persistence time to non-zero. And then we got very, very different behavior, as you might expect. That if you zoom in on different edges of this viable rim of the tumor, you'll see these green plumes of cells kind of invading into the red tissues. So this is kind of neat for two reasons. First of all, those green plumes in our model if we hadn't differentially visualized them, would be an invisible structure. We would just never see it. And so I think that's kind of the novelty here, is that you have a beautiful experimental system that makes structures visible for the first time. And we as mathematicians and modelers have the opportunity to explain these newly visible structures. And so our model predicts with this very basic set of hypotheses that you get the formation of this green plume. The other thing that's interesting is that if you were to just look at a static snapshot, it would be very tempting to say, hey, I have a clump of green cells that are doing something together. Boy, that sure looks like collective cell migration. But what we can show is that there is no communication between those green cells. They are not coordinating in any way whatsoever. All that happens here is that by stochasticity, one of the green cells finds a little bit of a mechanical weakness and squeezes through. And that makes that mechanical weakness a little bit weaker still, so that more green cells can follow through. As you get this small perturbation grows into a plume that invades into the red tissue. And then if the persistence time is long enough, then those green cells can punch all the way through the red material and escape. So that's kind of cool that we can predict, that as a function of phenotypic persistence time, of how long you keep your old migratory phenotype, you can predict the emergence of an invasive phenotype. And another cool thing is we simulated this first and got these green plumes and said, oh, we're really sorry, they're doing this weird stuff. And then we showed it to Danielle and said, oh my God, that's exactly what we see in our experiments. We hadn't seen that. So that came completely out of the model, and these microstructures were validated by the experimental system. And so that was quite neat. Again, so you have this novel imaging system that can show you previously invisible structures, and then you have a mathematical system that can explain those new structures. And so it's a potent combination of math and engineering, of modeling and engineering. And I'd like to point out that anytime you see one of these QR codes, you can try this model yourself online on NanoHub. So we hope you do explore it. So one last kind of research-driven, I guess, example before we go into a live demo is I want to show you some COVID-19 modeling that we've been doing recently. This is not our work alone. This is work of a wonderful coalition of contributors from across 20 institutions. Multidisciplinary of virologists, immunologists, mathematicians, computer scientists, working together to iteratively, step-by-step, try to build a general COVID tissue simulator that will hopefully capture enough of the realism that it's worth using for research. So we iteratively built prototypes, and then every time we released a new version, we updated our preprint on bioRxiv. So our first prototype we actually built overnight in like 12 hours. We took one of our existing nanotherapy models and said, well, virus is basically a self-replicating nanoparticles. So we took our nanoparticle model and then made them replicate inside the cells and then release and diffuse in the environment to start infecting other cells. So that's our first prototype. And while we released that prototype, we started building our coalition. The version 2 model said, well, virus doesn't just stick to a cell and enter, it has to attach to a receptor. So we added some really basic ACE2 receptor dynamics to say that virus had to bind to receptor gets internalized, has to dimerize, internalize, releases contents inside the cell, and then can either be degraded or traffic back up to the surface. So receptor trafficking could potentially be a limiting factor, although we haven't found it to be the case. Version 3 started finally adding the immune response. This is where our coalition started to take over and really lead on it after we started the initial development. And so they built agent-based models of macrophage activation, secretion of inflammatory factors to recruit other immune cell types, and in particular CD8 positive T cells to do effector T cell attacks, and neutrophils to do mostly like an ROS-based attack. Version 4, though, realized that, it's not just about that local response. You have to have the systematic response too. So we had the local inflammation leading to recruitment of immune cells, but then they go to a lymph node where we had an ODE model to model the ramp up of CD4 and CD8 positive T cells and the creation of antibodies that could come back into the tissue. And then version 5 model, we added neutralizing antibodies. And so little by little, as we added different aspects to the model, we were able to more and more model the course of the acute and the adaptive, the innate and the adaptive responses. So let me show you kind of an example of this model. This blue sheet here is like a layer of epithelium. Virus particles land randomly on us, so you can see little patches of infected cells. So the yellower the cells get, the more infected they are until they die because of the infection. So you see a black cell over here that's apoptotic or pycnotic or the other death mechanisms. Then the shades of green are different, polarizations of macrophages that come in. And as they start munching on dead material, they start increasing their secretion of inflammatory factors that it's going to recruit in dendritic cells to start sampling the antigens in the infected cells, and then they leave to the ODE model and drive the ramp up of the CD8 and CD4 T cell army, and of course the other cell types and the neutrophils, no, not the neutrophils, sorry, the antibodies that come back to the local model and start cleaning up the infection. So let's watch the dynamics here. So you have this infected cell, you do have some damage as the infection spreads, but now the immune system is kind of coming in and clearing out the infected cells. And well, you do have a lot of initial damage, but the infection is halted. You can actually plot the viral count, and it peaks here for several days in this particular model, and then it kind of goes down. And now we have, a damaged tissue, but the infection has been halted, and we do have a fairly angry immune system wandering around here. But this version of the model was sufficient to completely clear a COVID infection. So that was good. So we start with a naive immune system, and it does adapt and fights the infection. Then here's the cool thing. There's some memory in the system. If you reset the tissue now, say, now let's go to a different patch of tissue, but with the same immune parameters now that have been trained by the previous one, and now introduce a second exposure to the virus, and it's protective. You have a little bit of tissue damage, but the immune system quickly brings it under control. So you can show that that prior exposure, or let's say if you've done an immunization, is protective against subsequent exposures. And so that was actually quite cool to see these pop up out of the models. So what I'd like to do now is transition and kind of show you the live cloud version of PhysiCell and show you how you can build some of these types of models online. And then next week, if you come to if you stream some of our workshop, we'll show you how to extend with the C++ version of PhysiCell to add the dynamical rules to the cell agents. And so what we're going to do is we're going to build a basic immune model here. We're going to have bacteria. They're going to aggregate by a quorum factor and secrete that quorum factor, and they'll be randomly motile. And the macrophages are going to be attracted to the bacteria and are going to phagocytose dead cells, and they're going to secrete a pro-inflammatory factor like IL-6. And then neutrophils are going to be attracted to the pro-inflammatory factor, and they're going to phagocytose or eat live bacteria. So this is something we can actually build completely online or in PhysiCell without any C++ at all for this kind of model. So here's kind of our checklist. We're going to go to the online simulator. We're going to set up our domain. We're going to set up the diffusing substrates in our problem. We're going to add the bacteria and make sure it works. Then we'll add the macrophages, make sure it works. And then we'll add the neutrophils, make sure it works. And I'm going to keep a real close eye on the time. And if we have to cut it short, we'll just jump to the end result. But you can review these slides to get all the middle steps. So first thing we're going to do is we're going to go and click on this link. And hopefully, you will see, do you guys see my web browser over here?
Yep.
Oh, good. So it's switching between windows. That's good. So we're going to start by just clicking Launch Tool. I'm already logged in. So, and I'll give kind of a navigation through this app. So this is basically a virtual machine running in San Diego. And it will spin up in just a second. There we go. So what you see as this loads up, there's a variety of tabs that you interact with the system. The About tab just kind of gives you some basics about how to use it and where to go find more detailed documentation. Configuration basics is where we're going to set our domain. Microenvironment is where we're going to set up our diffusing equations. Cell types, we're going to introduce the bacteria and the other cell types. Other user parameters over here, user parameters, and then we can actually click and run in the browser and then visualize when we're done. So let's go ahead and start setting up this problem. So the first thing we're going to do after navigation is we're going to start setting up the domain. So I'm going to leave the domain alone for now. I'm going to go from -500 to 500 in X and Y in a very thin slice of Z. So it's like 2.5D. And I'm going to change my time down to 1440 minutes or 1 day of simulation. And I'm going to save my output every 20 minutes. So let's go back to the window here. I'm going to leave the domain alone. I'm going to leave on the virtual walls. That basically keeps cells from leaving the domain. And I'm going to change my maximum time to 1440. So now I've set up and I'm going to save every 60 minutes. So that's all good. And I'll go click on microenvironment. That'll be the next thing we're going to set up. So we've set up our domain, kind of going through our checklist here. And now we're going to start setting up the microenvironment with our diffusion equations. So we're going to go in the microenvironment tab. And I'm going to double click the substrate that's kind of like the default name. And I'm going to rename it to quorum factor and start setting some parameters to give me a 100 micron, not millimeter, sorry, 100 micrometer diffusion length scale. and I'm going to turn off the Dirichlet conditions. So it's going to be 0 flux conditions and a 0 initial condition. So until the cell secretes stuff, there's going to be nothing there. So let's go ahead and go into our simulator. I'm going to double click this so I can rename it quorum factor. So you are allowed to use spaces in most of these names. I'm going to change my diffusion coefficient to 1,000. I'm going to change my decay rate to 1. So if you take square root of diffusion over decay, and that should give you a 100 micron diffusion length scale. My initial condition is 0, Dirichlet condition is 0. We're going to turn off my Dirichlet conditions to make it a 0 flux condition. And it wouldn't hurt to, okay, to say, oh, so yeah, we'll do that. Let's see, so that's one of our diffusion equations. Now we're going to make IL-6 our pro-inflammatory factor. And for now, we're just going to basically just copy this. So copy. I'm going to rename this to IL-6. And I'm going to leave the same exact parameters for this. Okay, it's just for this. And let's see here. Do we have any other diffusion equations? No, we're done. So we've added all the diffusion equations. And now we're going to add bacteria with the quorum factor and make sure that they aggregate. And we're going to test this thing out. Okay, so now the first thing we do is we're going to go to the cell types tab. And you see that there is kind of a default name, just called default, not particularly interesting. So we're going to double click that and rename it to bacteria. And then we are going to choose a different cell cycle representation and give them the life cycle model and set a cell cycle duration of about 600 minutes. So let's do that. So first of all, I'm going to rename these to bacteria. And then I'm going to go to the cycle model. And there are a variety of built-in models in PhysiCell for cell cycle representation, but we're going to use the very simplest one where the cell is basically live or dead. And so I can represent my cell cycle as either a directed graph where I say how long do you spend in each phase on average, or the transition rate between phases. So let's just use the durations for simplicity. And we'll do a fairly slow cycling bacteria that spends on average 600 minutes in the cycle before it divides to another cell. So just a really, may not even be the most realistic parameter values, but will be ones that will show you the concepts. So we've set up cell birth by changing that. Let's see here. Now we should set the death rate. And what I'm going to do is I'm going to set a death rate that's on the order of 1/10 of it. So here's apoptotic death rate and necrotic death rate. I'm just going to set my apoptotic death rate here to 0.0002. That'll be enough to just kind of get some background death in the bacteria. So you have mostly proliferation, a little bit of death, so we'll actually see something. Okay, so we've set up death. Now we're going to modify the mechanics. And so what I want to do is to say that these are not a sticky species of bacteria that I'm going to model. So I'm going to set the cell adhesion strength to 0. So I'm going to skip volume. Now this is an unrealistically large volume, so we could change that later. But let's move on to mechanics. And I'm going to turn cell adhesion off. So I'm going to set that to 0 and leave the rest alone here. And now, so we've set the birth rate, the death rate. We made them non-adhesive. Now we need to start turning on motility. So we're going to make them chemotactic. And so we're going to go to the motility tab, and we're going to set the speed and persistence time to 1 and 5, respectively, bias of 0.5. And so this basically is for a bias random walk. They're going to move, they're going to choose a direction, move on average for 5 times for that persistence time, and then choose another direction and keep doing that. And then the bias says how much we prefer the quorum factor gradient compared to just picking a completely random number. So if my bias had been 0, I'd have Brownian motion. If my bias is 1, I have deterministic chemotaxis. And so there are a couple of gotchas. I'm going to go over here. First of all, in motility, we need to make sure that we turn it on. So we're going to enable motility. I'm going to set the speed to 1, my persistence time to 5, just as we have in our notes. I'm going to leave the bias at 0.5. So right now, with this enabled, the cells are motile, but they still need to have a direction. So I need to enable chemotaxis. And then I can use this drop down to choose which chemotactic factor I listen to. So I'm going to listen to quorum factor, and I'm going to move up the gradient. So we'll click towards. And that's it. I've got chemotaxis built into my model now. Now I'm going to go to secretion. We need to make sure that the cells are secreting the quorum factor. So I'm going to go to the secretion tab over here. And I can choose between the different things I'm modeling, but let's work on quorum factor. And I'm going to make sure that they are secreting it. So I'm going to just pick a nice high rate of 10 so that there's a lot of quorum factor being secreted towards a maximum value of 1. So when the cells when the local concentration reaches 1, they'll slow down their secretion because it's like proportional to that difference. So now we have secretion of the quorum factor. And let's see, is there anything else? Well, that's all we want the bacteria to do, right? We want to have them a little bit of birth, a little bit of death. We want them to have no stickiness. We want them to have secrete a quorum factor and migrate chemotaxically towards that quorum factor. So I think we're good to go. Let's increase the number of cells to 100. So we'll go here to the user parameters, and we see number of cells over here is an integer variable. We're going to set the initial number to 100. And we are ready to go. So we're going to click over here to run and click the run simulation button. And just give it a second. It should start putting your standard C output in this window over here. Yep, there we go. Now it's running. This should be pretty fast. We're already, the time that I'm blabbing, this will almost be done. Now, if you have multiple cell types, you may want to click on the legend tab, and it will give you a legend of all the different types of cells in your simulation. But now let's go over here to plot. So if I click like this arrow, I can see my random distribution, my initial placement of bacteria. I can click this button to advance through one frame at a time, so we can see the proliferation and the aggregation. And we can also then plot substrates at the same time. So I can click here on substrates and then do a contour plot of the quorum factor. So this drop down menu, let's choose. You can see how the quorum factor is clustered right around the cells, exactly as you would expect. Here's my IL-6. There, nothing secreting IL-6, so there's a flat gradient. And now if you want to play, you can click this play button, and it will advance through all of our simulation frames and do kind of a somewhat choppy simulation, or animation, but it will show you all the steps in our simulation. And so we simulated one day. We have the bacteria proliferating. We started off with 100 agents. Now we have 970. Then they're aggregating just like we expect. So I'd say that the first test here is looking pretty good. And this is, I think, what I would suggest in general is build your models step by step. Don't build everything and then click run for this first time, because something will probably be broken. And it's better just build it up one piece at a time. That also lets you explore the impact of every new piece that you add. It's like additive manufacturing. So let's go back now and see what we ought to do. Going through our checklist, we added bacteria. Now it's time to add our macrophages. Remember, these need to wander around by chemotaxis and be attracted to the bacteria. And in fact, there is literature that suggests that macrophages can chemotax towards quorum factors. And we're going to have them phagocytose dead cells in particular. So let's go to the cell types. We're going to take the bacteria and just copy it and make them to macrophages. And what I want to do is I want to turn off cycling. I want to make sure that macrophages don't cycle so I can just focus on their work. So I go over here to cell types. I'm going to click on bacteria, click on copy, and now I've got a new cell type. I'm going to double click the name so I can name it macrophage. I thought it was. There we go. No need for the macrophages to shout. There we go. So we have a macrophage. And go over here to cycle. And actually, if I pick transition rate representation, that's maybe a little bit easier. I'm going to set the transition rate for macrophages to 0, so they're not cycling. I'm going to click here on death and set the death rate to 0. So we're going to have like a fixed population of macrophages in this particular model. So we've gone to cycle and death, and we set the transition rates to 0 to have no birth and no death. And now we're going to go to mechanics. I want to make these macrophages able to sense over larger distances. So I'm going to set the relative adhesion distance to 1.5. So let's go here to mechanics. So we're going to not model cell adhesion with the macrophages, but we are going to increase the distance over which they can penetrate and sense to 1.5 times the cell radius. Okay, what's next? So now we should go and we should make sure that these macrophages are not secreting the quorum factor. Remember, we copied the bacteria, so we picked up all those parameters. So we want to make sure we turn off secretion of the quorum factor and turn on secretion of the pro-inflammatory factor. So let's go here to secretion. And first, for the drop down, I'm going to adjust parameters for quorum factor, and I'm going to turn my secretion rate to 0. And I'm going to choose from my drop down my pro-inflammatory factor, IL-6. I'm going to say, well, let's make sure that my macrophages are secreting IL-6 towards a target value that's non-zero. This is a gotcha. This hangs people up sometimes. You want to make sure that you have a non-zero target value, so they're going to keep secreting until they reach a non-zero value. So now we have macrophages are not being born, not dying. They have a little bit longer of a sensing distance for mechanics, and they are secreting the pro-inflammatory factor and not the quorum factor. Okay. Oh, and then the most important thing for macrophages is that they better eat something. So we're going to go over here to the interaction tab and choose macrophages. And I'm going to set the dead cell phagocytosis rate. So in our code, actually, PhysiCell, they can make, they can have rules based upon the cell type. So if I run up and touch some cell, I can test to say, am I alive or dead? And if that cell is alive or dead, let's say it's dead, I can say, well, it's my rate of phagocytosing dead cells. And so here, for our macrophages, I'm going to set a rate of 0.1. So this says that, 1 over rate is like your mean waiting time. So 1 over 1.1 is 10. So I have, on average, my macrophage is going to spend about 10 minutes in contact with my dead cell before I phagocytose it and move on. And then it acquires all the volume in the mouse, it has to digest it and moves on. You can also set things like phagocytosis rates of live cells, which we'll do later. But I think we're good to go here for macrophages. And so let's test it. So let's go back to run, click run simulation again. And we need to see this kind of reset, right? Make sure it's really running. Oh, there we go. Now it's running. So I'm going to go click on this to go back to the beginning of the simulation. And I want to click the pro-inflammatory factor just to make sure that it's doing what we expect. So I'm going to go forward a couple of frames. Oh, notice, right? We have a new cell type. So you go to legend. Now we have bacteria and macrophages. So now you can see, right, that the pro-inflammatory factor is clustered right around those macrophages like we expect. And those macrophages are attracted to the quorum factor, so they are co-localizing with the bacteria. I'm going to turn off the substrates so we can see things a little bit more clearly. And maybe if we luck out, we'll see them. So here's some dead cells. But I guess because the bacteria are primarily attracted, the macrophages are primarily attracted to the live bacteria, they're kind of stuck. We could change that, by the way, but let's leave it simple for the demo. But, we've, in not much time, like 15 minutes, we've built a simulation from the ground up that has a couple of diffusing factors, a couple of cell types, and I think we have just enough time to finish the model. So we tested them. Oops. Oh, stupid Macintoshes and their backward scrolling. Okay. There we go. So we added macrophages and tested. Now we need to go to neutrophils. So we are going to copy the macrophages and we're going to rename them to neutrophils. And then we want to make sure that their motility is that they're chemotactic, moving towards IL-6 instead. So let's go back to cell types, click on macrophage, copy it, double click over here to rename it to neutrophil. Okay, and so let's kind of click through. They have the same cycle, no cycling, no apoptotic death. I'm going to leave the volume and the mechanics the same for now. And motility, the only difference is I want them to be chemotactic towards IL-6, towards that inflammatory factor. And that's it. Secretion, I don't want them secreting anything. That's not the job of these neutrophils. So secretion of IL-6, we're going to set to 0. And then in the interactions, I want them, they don't phagocytose dead cells, but they are allowed to phagocytose live bacteria. And so you can see here in live phagocytosis, you can scroll down and on a cell type by cell type basis, say how often I eat, what's my rate of phagocytosis in the different types. So I'm going to pick bacteria and say that neutrophils, so I have neutrophil selected, neutrophil can phagocytose live bacteria, and I'm just going to pick a rate of 0.5. So that says that they spend 1 over 0.05 or about 20 minutes on average in contact before they phagocytose something. Oh, one last thing. I meant to actually make my macrophages a little bit less deterministic to get a little bit more mixing in the system. Let me go back to cell types, go back over to here to motility, and I'm going to set my migration bias for macrophages a little bit lower at 0.25, just for the fun of it, I guess. But otherwise, we should be good to go. Let's double check our checklist here. We define neutrophils. We make sure that they're migrating up chemotaxis of IL-6 instead. We turned off the secretion of the pro-inflammatory factor for neutrophils, and we set up the interaction to make sure that they're eating live bacteria instead of dead bacteria. And we're ready to run. So let's do it. So click Run Simulation and give it just a second here. Should reset in just a moment. Okay, now you see the scroll bar, so you know it's running. Let's go over to plot and go back to the beginning. And so now you see that third cell type sprinkled in. So if I go to legend, I can see my third type is neutrophil. And so let's just advance a few frames. So we have the red macrophages should be chemotaxing towards the bacteria, the gray ones. The bacteria are aggregating in colonies like you expect. The macrophages are, oops, went all the way to the end. The macrophages are attracting the neutrophils, and you should see them now, you should see them eating the live bacteria. And if we give them even higher rate, they might be more successful. But think about what our initial population was. Our initial population is 300, 100 of each type. By the end, remember the original simulation, we were over 900 agents of just bacteria. So if there had not been any success in predation, we would have expected this to be easy, 900, 1100, 1200. So this is slightly reduced now with those neutrophils and they're eating the live bacteria. So it seems to be working. So if I went here back to cell types and made the neutrophils have an even higher live phagocytosis rate, let's say a factor of 10 higher, we should see some very different behavior. So let's go here to run one more time. These are really fast, like 20-second simulations. So we'll click run simulation. And we should see this reset in just a moment. Plot, let's go back to the beginning frame. And kind of cycle through. So we see them kind of catching up. And you should see them attacking the colonies. Let's just click play now. Oh, we only made it to 20 hours here. Didn't seem to have had much impact, so that worries me that the parameter value didn't take. Sometimes the cloud version could be a little bit laggy, and you can click on something and it doesn't seem to actually see. If I clicked here and then click back, I don't think that the macrophage, the neutrophil phagocytosis actually got accepted. So I'm going to make sure I click on a few fields. The desktop version of this runs a lot better, unfortunately. Okay, so now we definitely have a phagocytosis rate by the neutrophils. So just want to make sure you kind of click something else and then go back and make sure that value transmitted. Good. Now San Diego has the parameter value. So I'm going to click run one very last time. Only takes like 20 seconds, plus lag to start it, I suppose. And now it's definitely running. So I'm going to go back to the beginning. Advance a couple of frames. And we should see something very different. Yeah. Notice that the neutrophils are getting bigger because they're acquiring the volume of the things that they ate. So I think we definitely see something going on here. That's good. So let's click on plot and just play the simulation. And we have neutrophils going and hunting down the bacteria. The bacteria is still trying their best to proliferate pretty fast. So they really can't completely keep pace here. But we can definitely see the model in action here, and the final cell count is definitely lower, so it's working like we expect now. So that is a bit of a quirk with the online version of the code. That sometimes you click something in the interface and it doesn't quite take and transmit all the way to San Diego. Another good reason to do this iteratively, to kind of test one thing at a time and make sure everything you expected is actually working, and then you can kind of see it going. But there you have a complete example actually of building a basic immune model with multiple cell types doing different types of immune behaviors. And we will be walking through that next week if you want to learn more about it. So thanks very much for seeing it. If you click this barcode, you can actually run through the whole example, but I think Veronica is going to send you the slides, and we'd be very happy to see you in our workshop next week. So follow us and we will send you information on the Zoom calls. And I'm very happy to take questions.
Thank you so much, Dr. Macklin. That was an excellent presentation. Really love the live coding. And I really appreciate your suggestion to code it part by part and test how changes are affecting the model. I think that makes it a lot more digestible and easy to validate. Does anybody on the call have questions that they'd like to ask? Looks like Naveed has a question.
Yeah, thank you. Dr. Macklin, it was a wonderful talk. Thank you for it. And I've actually worked on hypoxia before, specifically for breast cancer. And I mean, something we share, I was actually also part of the Digital Twin project, the UMass branch of it. But yeah, it gave me time to think about this. I've worked on this both for atherosclerosis and cancer. So it seems like it's a very dimension dependent kind of problem. And given that when we were working with patient specific data, it seems like cancer can get really large. Like it's initial from its initial dimension to its like final dimension can be very, very large. I don't know if this was a choice you made because of I don't know, the time you had to show us the simulations, or have you tried like letting the simulation go a little bit farther so that the cancer like gets within its like realistic large dimensions? And if so, like how does that affect the I don't know, the plumes or the necrotic region or the hypoxia or things like that?
So those are really great questions. And of course, you look in a mouse, you look in a human, and these tumors are, centimeter scale, 10 centimeter scale, which is now no longer feasible for most agent-based models, including ours, even when you put them on HPC. What I would argue is when you get to that large of a size, you may want to be looking for other approaches. But the thing that I find, and actually here, I'm going to approach dimension from a couple different aspects. First of all, there is dimension in terms of 2D versus 3D. And there are some problems where that dimensionality actually really matters a lot. The classical example would be like angiogenesis. If you have a bunch of cells migrating, leaving a trail of vessels behind them, and they need to anastomose or form loops, in 2D, it's inevitable because you cannot topologically wander very long until you hit a previous trail of cells. So like anastomosis in 2D is guaranteed and impossible to avoid. In 3D, now you have the same thing where you have migrating cells that have to hit a line in 3D, which is almost probabilistically, much smaller. So you need active guidance to do it. So I mean, there is an example of a problem where 2D versus 3D matters. And a lot of these where you're driven by so what we often do is we'll build a model in 2D and then test in 3D, make sure that the lessons still hold. And quantitatively, they'll often be different because you have differences in gradients and densities and mechanics, but often qualitatively they tend to hold. So that wasn't your actual question, but I want to make sure to address that aspect of dimensions also. The other aspect is just in terms of scaling. This is order n in the number of cells, so it doesn't blow up quadratically like a lot of agent-based models. So that's good news. So theoretically, if you have enough resources and patience, you can simulate as big as you want. The question, though, is it worth it to do that? What we find is that once the tumors reach, several hundred microns in size, or even up to millimeter size, most of the dynamics have already emerged. That you have a clear existence of viable structures and of necrotic structures. The viable rims tend to start converging towards a characteristic thickness. And even the fractions of cell types doing different things emerge. And so what you tend to have is that after you reach a big enough size to really establish a lot of the gradients and the patterns and the population dynamics, they start kind of getting to something looking more like a steady population state. And so you can run them a long time to get things like evolution. That's different. But in terms of the more basic interactions and signaling, you'll often learn what you need to learn after a few millimeters in size. And that doing it bigger is just doing it bigger. In terms of clinical predictions, it's a different matter. And I think what you would want to do, and say if you're making like a digital twin, for example, and this is something we're thinking about a lot, is use the agent model to assimilate your data and calibrate, and then use your agent model to train a continuum model. So you can kind of transfer the data and the knowledge from biology and pathology and genomic scale measurements up to agents, use those to simulate long enough to get those long-term dynamics, and use that to train a continuum model that you use for long-term clinical predictions. But
yeah, I personally work with continuum models and
I'm sorry? No,
I personally work with continuum models, and hypoxia has like adds numerical instability to continuum models as well. It's like a tricky thing to work with, because if there's like hypoxia and it has a dimension-based kind of like action on your model, then it can like cause heterogeneity that is so drastic, like the gradient drops so fast that like it changes like the size of, I don't know, your mesh,
so yeah, we wrote models like that 15 years ago, but they're wrong. there's no way that's biologically plausible that a cell changes behavior and instantly you have a humongous change in your gradient. There's probably more subtle, I'm sorry, wrong is too strong, but they seem biologically implausible. That we ran into this when we wrote with like agent-based, not agent-based, but like continuum and moving boundary problem style problems, where you say, solve your tumor, look for the contour of a specific value, say that's where you're hypoxic or necrotic. And then the necrotic cells have a different uptake rate than your normal cells, which means you have a big, discrete, sudden change in behavior, which means that all of a sudden your contour moves. So you get like this flashing behavior in your tissue. And that's often shows you that there's something wrong in your model itself, in your formulation, and some of the biological realism, if you're getting numerical artifacts coming out like that, or it's a numerical artifact. But we ran into that and solved that about 15 years ago on our continuum models. Generally speaking, if you have stochastic individual transitions and more gradual changes in behavior, you tend not to see those artifacts emerge. But that's correct. Anything like that should really send off warning signs to you that you may need to revisit your biological hypotheses. If a small change in cell, if a change in cell behavior all of a sudden radically changes your landscape, which radically changes your behavior again. I would worry. We worry too. That's why we just simplified the model to just have it like a constant necrotic core. And not but you don't need a constant necrotic core either. I mean, you can, if this emerges gradually, you tend to get something a little bit more realistic. And this is actually a much easier problem to solve within the agent framework than in the continuum framework. Yeah, you're right. It brings back old memories now that you bring it up, right? Because that was actually something we faced, is we modeled, the viable rim has one uptake rate, hypoxic has a different uptake rate, and necrotic has a different uptake rate based on the current location of the contours. And that would cause problems. And actually, the way to solve it is to build some hysteresis or memory into your model, that the cells need to remember if they were dead before and stay dead. They need to remember if they're hypoxic before and stay hypoxic, so that you use the contours not as an instantaneous change, but the contours to tell you who should be added to the newly hypoxic population. And if you model that, then it works. Which is actually what's happening in our agents, is right, that they remember their background, so they're not instantaneously changing states to and back, because basically you had resurrection of cells if you have it based upon the contour. And so the solution to that, and if you look in my old papers from like 2003 or so, is to model the necrotic population as a separate level set function and just add new material to it based upon the current values. But then it can't revert back.
I see. Thank you so much. That was a great explanation. I appreciate it.
No problem. It's a real challenging problem. It drove us absolutely crazy, especially for these. I can imagine. Yeah, it drove us absolutely nuts. Yeah.
I want to give Matthias a chance to ask a question, but really quickly, Dr. Macklin, is the hackathon still open? Registration for the hackathon, or just the tutorial?
That's a great question. Unfortunately, we can only support so many people on a hackathon because it's very one-on-one. That we form teams and kind of co-mentor. So the hackathon, we have to limit to about 30 people, and we sent those invitations out quite some time ago. But keep our eye on us. We'll probably do it again next summer, and we'd be delighted to have it. The workshop, however, is completely open, and we'll stream it on Zoom. And all the materials that we create for the workshop will be made publicly available on YouTube for the videos and on GitHub for the slides and the code. That said, I might have 1 or 2 slots available if someone wants to come. The biggest criterion, like if someone came and like you're really available, like you can hang out in the afternoons and the evenings to really be a full-fledged, functioning member of your hackathon teams, I'd probably override my number and let you in. So send me a note if you can really commit, because that's exactly the kind of person we want in, and we'd be delighted to let you in. So I can like, I can tweak the numbers a little bit. I can't get another 30 or 40 people in. But if you really want to do it, please come talk to me. Yeah, we'll let you in. Yeah.
Thank you. Matthias?
Yeah, great talk. I have a quick question about using the initialization of this 3D structures. Can you use like image data from fluorescence microscopy or like segmented data and basically initialize your 3D structures in the simulation from that?
That's a beautiful question. This thing that's the kind of thing we would love to have our hackathon participants build in particular. is that now that we have kind of this computational core, we need to build that ecosystem of things that help you initialize models, things that help you calibrate them, things that help you validate and visualize and analyze them. And so that's kind of the next step, I think, in our community. That said, we can import things from microscopy, but we have to write an importer. One thing that the code can do is you can give a list of cells as like a comma separated value spreadsheet, X, Y, Z, and ID. And the ID will say, oh, I'm cell type 3, I'm a macrophage, I'm cell type 4, I'm a neutrophil. And so if you write that list, we'll import all those cells, place them where you told us to place them, and then choose the right kind of cell type to apply the cell definition and go from there. So we can do that. If you want to do something fancier, like let's say I have a bunch of cell positions and maybe I know if they're cycling or not, we'd have to write an importer to do that, but that's something that could be done. But it would have to be pretty manual and tailored to your particular data. And I think it's it's an area that's ripe for advance. Other measurements, like if you have force microscopy stuff, oh my God, that would be awesome, because we have not been able to really rigorously model the individual cell forces like we'd like to. We've had to kind of model them as kind of phenomenologically saying that we know their interaction distance. We know that we want to make them pretty repelling so they don't overlap too much. And then we know we can measure, at least in steady tissues, the mean cell density. And that mean cell density can be translated to mean cell spacing. And then we use that to balance our forces, saying that, you know, adhesion and repulsion should balance at that characteristic spacing between cells. And that's how we calibrate our mechanics right now. But if we had direct measurements, that would be really, really fantastic.
Yeah. Good question. And can you restrict geometries? I'm mainly interested, like, in having restrict the geometry space, like giving vessel space and then having regeneration of cells within this fixed vessel geometry and flow equations through the vessels and stuff like that. Is this possible, or is this too far away from?
We can do that to an extent in two ways. One thing we can do is, you saw that code for the virtual walls. It's basically using a distance function to add a little bit of repulsion if you get really close to the boundary. We can choose different geometries for that virtual wall to add things kind of interior to the domain. Now, with a caveat is that that's mechanically restricting, but it's not restricting diffusion right now. We can, though, also apply Dirichlet nodes within the domain to say that maybe there's like a region that's kind of like the far field tissue outside of that boundary. So we can actually kind of start approximating something like that. That's actually how we used to model DCIS, or Dr. carcinoma in situ. So that's one way to do it. You can add these distance functions as a way to specify a complex geometry. And actually we've done that a little bit. It's really fun. I take like a black and white bitmap that I draw in Microsoft Paint, import it, and use that to initialize a distance function. I'll have cells wandering around in little weird looking regions. So that's something that can be done. The other thing you can do is there is a Boolean parameter you can set on a cell aegis that says is movable, which is usually true. And if you set that to false, then what happens is that agent can impose forces on other cells, but it cannot be moved. And so you can create structures out of those to form rigid barriers. So that's the other approach that people can take for that.
Thanks a lot.
So you basically have dummy agents that act like walls.
Lucian, if you could ask a quick question, sorry, we need to transition to Jonathan.
Yeah, I know. So my quick question is just that, why didn't you already have a viral model before like COVID hit? Like it seemed like you had a lot of basic viral things. Like I'm surprised that something like that didn't already exist. Is it just, yeah?
Because time is finite.
Yeah, okay. But no one else had made one either,
we just couldn't need it in any of our particular problems up to that point in time.
And no one else did either, or no one was using
other people might have built them here or there. The problem is that there is no standard representation for agent-based models, and I know the SPM people say there is, but there isn't.
No, I don't think we just say that either. But yes.
Yeah.
No,
okay. Well,
I don't think we say that either.
It's been like for years since we've been to Combine. Yeah. Right now, basically, there's no direct way to import a model from one framework to another. And we're trying to build those kinds of rules, but there are a lot of gotchas. Even like we support SPML, but with like an asterisk. I think the easier way, more correct way to say it is that we support ODE models that are written in the SPML language, not that we support all SPML models. And because we need to have an interface between the inputs of that model and things that are in our environment, and we need to have an interface between the output of that SPML model and our cell behavioral parameters. And we have an interface that we developed, but that means that that's like a special class of SPML models and not all of them. So you can imagine that everyone's kind of doing that. And there's no agreed standard that allows you to say, take the viral replication model from Ted and go put it into the model from Bob and Alice.
Yeah.
It's too bad.
Right.
It needs to be built, but money. So,
yeah. All right, thanks.
It's a great question.
Thank you so much again, Dr. Macklin. That was excellent.
Thank you so
We're going to
much.
transition.
I will stop sharing my screen and try to